Level 1, that is targets that can be used to screen for likely presence of GMOs corresponding to words or simple phrases. With these targets, we may be able to sort products likely contain GMO from products not containing GMO. Level 2 are fused target, like the constructions I already showed you. These provide evidence of presence of GMO, but they are still not really reliable when it comes to identifying and perhaps quantifying. And that is where we use event-specific targets, that is the level 3 corresponding to inserts fused into the original text and that unique motif. These targets give the ability to discriminate authorized and unauthorized products. The application of methods and results related to the method and technology developments include methods now part of various international standards, recently conceptualized also for microbiological applications, methods routinely used in numerous private and enforcement laboratories worldwide, improved knowledge of characteristics of GMOs, which have improved the basis for risk assessments. And projects have also provided very important contributions and input to the development and revision of EU regulations, in particular the key GMO regulations 1829 and 1830-2003. It is, for example, now required that an event-specific method must be provided by the applicant prior to authorization of a GMO in the EU. So science has made significant achievements, and that's not bad. But the story does not end here. With an increasing number of GMOs potentially in the food supply chain, it would be nice if we could detect all the GMOs in one test. It could speed up and reduce the costs of testing. So development of multiplex methods has been a high priority of the laboratories. One example is a PCR microarray, now a commercial product that was successfully validated in a so-called collaborative trial among a large number of laboratories as part of the CoExtra project. Several of the GMOs on the market now, in particular maize and cotton GMOs, are so-called stacked events. This means that they carry multiple insertions. Nearly 50% of the dossiers received by the European Commission since 2004, and we're talking about approximately 100 dossiers, 50% of those concern stacked events. So metaphorically, someone may publish a version of Hamlet with 2, 3, 4 or more new inserted sentences. In the CoExtra project, we reviewed the challenges posed by stacked events. I will not go into much detail, but essentially we recognize that from a labeling point of view, there are problems more likely to have a policy than a scientific solution. The number of GMOs developed and released around the world has been constantly growing over the last 15 years. The global trade is also increasing. Thus, a new problem has become apparent: presence of unauthorized GMOs. Let us roughly divide these into two groups. I think Eve already mentioned those. First, we have those that are products authorized in exporting jurisdictions but not in the importing jurisdiction. If a GMO is authorized in the exporting jurisdiction, we can take a few things for granted. It has been assessed for risk for human and animal health and to the environment prior to release, and it has been found safe. And a specific detection method for the GMO is available, at least to the authorities of the exporting country. Several GMOs in the EU regulatory pipeline fall into this group. They are unlikely to represent a true health or environmental risk. Often an event-specific method is available, and if so, the GMO is detectable. Thus, these are not a major problem. I think it is clear from what I have explained so far that detection is easy if we know what we're looking for. But what if we do not? This brings us to GMOs that are neither authorized in the exporting nor in the importing jurisdiction. There are several examples of unintended releases to the food supply chain of GMOs that have only been approved for field trials. In a transparent jurisdiction, that may still not represent a severe risk to health and environment, but it certainly can have severe socio-economic consequences, as illustrated by the LL601 rice case in the USA and current emergency measures on rice imported from China to the EU. Now, imagine that a GMO is illegally released into a product for export in a country outside the EU. It is not authorized in the EU, and nobody in Europe is informed about this release. This GMO is likely to be incompletely risk assessed. Furthermore, if at all there is a detection method for that GMO, the method is likely to be incompletely validated. Will we be able to detect that GMO? The answer today is only if the genetic modification contains one of the signature text elements of GMOs that we already know and test for. In other words, the answer is often no. Metaphorically, inserts like "never look on the bright side of life" or "always look on the right side of your wife" may both be detectable, but not "never hook up the right side of your wife," despite its similarities to the text from Life of Brian, and even less so a text such as "umakrinak manna sapisangilavut" that bear no similarity to anything we would expect to be a genetic modification inserted into our species or book. If a GMO is authorized in the exporting jurisdiction, sorry, that was wrong. So today the developers of GMOs have virtually unlimited sources of elements that can be used in novel genetic constructs. That was not the case 10 or 20 years ago, when most of the currently dominating GMO plants were developed. The detection tools we apply today, therefore, are likely to be of very limited use in the near future. Some of the GMOs recently entered into the EU regulatory pipeline are modified by inserting extra copies of DNA from the receiving species, such as potato sequences inserted into potato. These too are extremely difficult to detect unless an event-specific method is made available. It is therefore our recommendation that much more focus on GMO detection and risk in the future is directed towards the unauthorized GMOs in general and unknown GMOs in particular. Development of tools with potential for detection and characterization of unknown GMOs was initiated but far from completed in the COEXTRA project. While we wait for resources to develop the tools to detect the unknown GMOs, there is a significant and growing risk of presence of such a GMO somewhere in the global food supply chain. Given that it would be unknown, it would also not be possible to assess if it could do any harm to economy, environment, or health. Can we afford to ignore this? I have come to the take-home messages. We do have most of the tools we need to be able to control products for compliance with labeling regulations. The exception is products potentially derived from stacked events where the issue probably needs a policy rather than a scientific solution. We have made a lot of progress on cost efficiency and reliability of GMO detection methods, but there is still more to do. GMO laboratories all over the world now largely speak the same language, although there are some harmonization issues left. The major problems, in our opinion, are associated with the unauthorized GMOs, and in particular the unknown GMOs. Improved transparency on a global scale is undoubtedly a key issue to improve our ability to minimize risk. Unless developers and regulators all over the world provide information on GMO developments, field trials, etc., we are certain to be unable to detect the GMOs that will be developed today and in the future. Thank you. And
well, thank you for that. A fascinating and an original take on the whole process of genetic modification. I shall never hear Monty Python's Life of Brian or read Hamlet without thinking that.